Hi everybody, so today's lecture set will be about ribosomes and their role in translation, as well as ribosome structure. Just to remind you, we're talking about the second part of the central dogma, so how RNA is translated into proteins. The process is called translation, and the protein and RNA machines participating in the process are called ribosomes. Here in this diagram, they're depicted right there. You can see that they have that their bottom part, the smaller part of the ribosome, the small subunit is attached to the RNA molecule, and then the catalysis and the formation of the peptide bonds occurs inside the larger subunit. And they read from the 5' to the 3' direction. They read the mRNA and translate the mRNA into protein. Just to remind you, because I think it's important, here's a slide from the genetic code lecture set. Here I would like to remind you guys again about the different parts of the mRNA molecule and which parts are translated and which are not. And the fact that the promoter area inside the DNA is unlikely to contain the start codon. The area immediately to the promoter, but both in bacteria and in eukaryotes, the starting codon for the translated protein sequence will be located further into the mRNA molecule. So again, we have a 5' cap, which is basically an inverted GTP, and we have a poly A tail. All eukaryotic mRNAs will have them. If they don't have them, they will be degraded, because both of their ends are not protected, and riboexonucleases will be able to get in and destroy it. Then there's the 5' untranslated region, and there's the 3' untranslated region that are located respectively in front of the first starting codon for translation and after the last stop codon. So here's the picture. So there's the first ATG, the starting methionine, and the stop codon that tells the protein to stop translation. Tells the ribosome to stop translation. And you can see that the starting methionine is incorporated into the peptide structure as it comes off the ribosome, but the stop codon does not code for any amino acids. So the last amino acid here is the valin that the GTC codon was coding for. In the green, you can see the untranslated 5' region and an untranslated 3' region, so 5', 3', as well as the poly A tail, and none of that stuff becomes protein. However, the stuff in blue, the codons in blue, as translated from the ATG in triplicate codons, will all be translated into protein until the first stop codon is encountered, and that area of the mRNA is referred to as CDS, the coding sequence, or the open reading frame. All right, so what about the awesome machine that does the translation? Just to remind you guys, the difference in what the tRNA does and what the ribosome does. The tRNA is the adapter essential for the translation step. It is the tRNAs that allows to convert 3 codons into a particular amino acid. But the tRNA cannot, but it's the substrate in the reaction because it has changed. It comes in there charged with amino acid and leaves empty. It has lost the amino acid that has been attached to the polypeptide change. So in the translation reactions, the tRNA is the adapter/substrate because it's altered during the reaction. The ribosome is the enzyme. The ribosome is the catalyst that makes the reaction happen because it creates a particular environment to facilitate the formation of the peptide bond. The ribosomes are complicated machines. The large subunit of the ribosome, the 60S subunit, consists of about 50 proteins and then 3 different RNA molecules folded all in particular ways, while the smaller subunit that does the presentation of the mRNA template contains about 33 proteins and then one RNA molecule, the 16S rRNA, ribosomal RNA. In this electron micrograph, you can see hundreds of, probably thousands of ribosomes that are all these tiny little black dots coating the endoplasmic reticulum of our cell, but also found just hanging out in the cytosol as well. And the ribosomes are so large that you can see them in electron micrographs. Normally you can't see proteins, they're just simply too small. But the ribosome is such a large arrangement of proteins and RNA that they can be visualized under the electron microscope. And here the sizes are about 100 times larger than an average protein and even 10 times larger than the quite big RNA polymerase complex that is responsible for RNA transcription. A little bit about the way that this large and the small subunit of the ribosome are named. In bacteria, it's the 50S versus 30S subunit. In eukaryotes, it's the 60S versus 40S. The S stands for Svedberg units. Svedberg units is a way to measure density of something during a high-speed centrifuge spin. So the sedimentation rate of these was for the large subunit of the ribosome was about 60S, and the sedimentation rate for a small subunit of the ribosome was about 40S. And since people worked in ribosomes for decades by now, that's why they were originally named the 60S and the 40S ribosomal subunits, or the large subunit versus the small subunit. Same goes for the rRNA. The reason it's called 16S is because it sediments at 16 Svedberg units. Okay, so here is it's not really a crystal structure, but it's a true to the crystal structure representation of the ribosomes. Because they were so incredibly large, they're actually quite difficult to crystallize. And these structures were achieved by combining pieces of the ribosomes. So here you can see that the prokaryotic ribosome is a little bit less triangular, a little bit more rounded shape, like a little clamp. The top bit is the 50S subunit, the large subunit, and the bottom part is the 30S. In eukaryotes, it's the 60S versus the 40S subunit. And you can see here how much RNA these ribosomes contain. Protein makes up only 1/3 of the total mass of the ribosome, and you can see here that the space-filling bits are protein, and all the inside, and even protruding in various places on the outside, are the little noodle strings of the rRNAs. The rRNA, especially the 16S rRNA of the ribosomes, are very, very conserved. Not only is their nucleotide structure conserved, the way the fold is conserved, you can see that how few differences are there between the bacterial rRNAs and the eukaryotic rRNA, 16S RNA. And why is it so little change? Well, all ribosomes have the same job. That job has been optimized by evolution for millions of years. By the process of trial and error, we have all arrived at the most optimal structure of the rRNA for the ribosomal function. So, thus our ribosomal RNA is nearly identical to the bacterial ribosomal RNA. In fact, there are so few changes. The rRNAs are so similar to each other, and there are slight changes in non-critical locations that you can trace the relationship of everything that's alive right now based on the sequence of their 16S ribosomal rRNAs. It's a very common method of A, identifying a particular species, and B, establishing relationships between two different species. In fact, when people, for example, try to figure out what kind of bacteria live inside your gut, in your microbiome inside your intestine, the sequencing that will occur will be of 16S ribosomal RNAs, and it's the 16S ribosomal RNAs that will allow to figure out which particular bacteria are present in your microbiome population. So let's look a little bit more at the ribosome. As I said, ribosome is the catalyst of the polypeptide bond that allows for the creation of the polypeptide chain. There's 3 important binding sites for tRNAs inside the ribosome, and there's also a binding site for the mRNA, the template mRNA, inside the smaller ribosomal subunit. The sites for the tRNA binding are the amino acid side, so amino acyl side, the A side. Then we have the peptide, polypeptide site, the P site, and then we have the exit site, so the E site. And so this is how typically you see the ribosomes represented in textbooks, where there's like these 3 arches for the 3 sites. But this is the actual crystal structure of the ribosome, and you can see that the spaces are not identical, and that you can also hopefully appreciate from this diagram, and especially from this diagram, that the tRNA molecule is rotated as it is moved from the A to the P to the E sites. Especially the position of the 2 tRNAs inside the A site and the P site is especially important, because the 2 tRNAs are bent together just so that the area where the amino acid is, you can see it right here, are positioned really close to each other, allowing for an easy chemical reaction to occur there. Remember that both tRNAs that are sitting on the sites have arrived with activated amino acids. They're not just amino acids anymore, they're amino acyls ready to react and form a peptide bond. They just need to be positioned into close proximity with another amino acid in order with the carboxyl group of the growing polypeptide chain in order for the bond to form. So the actual peptide bond formation reaction does not occur in the A or in the P site. It occurs on the edge between the 2 sites. And you can see it right here, where the 2 tRNAs have been positioned very closely. Now, by the time the tRNA is then placed into the E site, it has lost its amino acid and it's just ready. It's no longer charged with amino acid, it's just ready to leave the ribosome. And you can see that there's a substantial opening for it to be able to leave it. So what I like about this diagram as well is that you can see that this is the newest tRNA that has arrived into this position. And it's still holding its own amino acyl group here. The tRNA in the P side has a polypeptide chain fully attached to it, and as the reaction occurs, the sites of both tRNA molecules will be so close that this new peptide bond will be formed between the carboxyl group of the second amino acid and the amino group of the third amino acid. Now, this tRNA here in the exit site was the tRNA that brought this first amino acid to the ribosome, and it's ready to be discharged. It has now discharged from the ribosome because it has completed its task. An important thing to realize about ribosomes is that it's the RNA that is the catalytic catalytic member of the ribosome. The proteins are not the ones performing catalyzing the reaction, but it's the positioning of the tRNAs by the ribosomal RNAs into this very tight configuration in the P side and the A side, so the 2 amino acids are close together, allows for the peptide bond to occur. So ribosome is a ribozyme. the RNA inside the ribosome is what allows, what the catalytic part of the ribosome is. And you can, so there's two ways that I can show you, prove this to you. So the first set of experiments was done a while ago, back in the 70s. And the scientists who were working trying to understand which protein inside the ribosome was responsible for catalysis of the peptide bond, tried to remove as many proteins as they possibly could, then figure out which protein was left and whether the ribosome was still able to catalyze polypeptide bonds. And they were pretty rough about it. So the first thing that they did is they added phenol. So phenol is a solvent. It will remove everything, all the hydrophobic proteins out of the ribosome, but leave charged proteins there. So with phenol, phenol would remove it. Proteinase K is a proteinase, so it destroys proteins, but not RNA. So proteinase K digestion would have destroyed all the proteins inside the ribosome. They've added that. SDS is causes the unfolding, the breakage of the hydrogen bonds between the proteins, holding them in their particular three-dimensional shape. So they've added even that, and still they saw a product. You can see that. So this is a product showing that the first methionine was incorporated. You can see that if you add the ribosome, but as well as SDS, which just has unfolded all the proteins, the proteinase K, which has digested the proteins, and the phenol, which has extracted all the hydrophobic, hard-to-get-to proteins from this 50S ribosome, is still able to catalyze the formation of a peptide bond. So it was the RNA, that was the real proof. It was the RNA that was able to catalyze this reaction, and not any of the proteins inside the ribosome. The other thing is that once the ribosome was crystallized, once the large 50S subunit of the bacterial ribosome was crystallized and its shape figured out, it was quickly realized that the closest protein to the active site of the ribosome was 18 angstroms away, which is very long distance in terms of being able to participate and catalyze the reaction at the active site. So you can see that the closest one is 18 angstroms, and the other closest ones are 26 angstrom here, and then the 39 angstrom right there. They're very, very far from the active site. But you can see how the active site is full of RNA. So, and you can, if you've seen in the previous picture that the ribosomal RNAs push the tRNA molecule so close that the two amino acids are only 7 angstroms apart, and then the peptide bond catalysis can occur. So let's look at that a little bit further. So this is, in the previous pictures 2 slides ago, you saw how they're pushed together, and this is the actual distance that they're pushed together. They're only 7 angstroms apart, and because both the amino acids not pictured here have already been activated and now no longer amino acid but amino acyl in this position, they're very eager to react with each other because they have been placed very close to each other. So electron exchange can occur and a peptide bond can be formed. Another thing that the ribosome does, aside from positioning the 2 tRNAs in the A and the P site close together, is assure that the codons are read in triplicates and there is no sliding. So the 30S subunit of the ribosome, or the 40S in eukaryotes, introduces a special kink in the RNA template, preventing these the tRNA here from binding base pairing to the incorrect nucleotides. And it's the kink of the mRNA that also will position the tRNA just so that it can start reacting with the amino acid in the P site. Another thing that happens that the ribosomal subunits do in this case particularly the smaller subunit that is the template, the mRNA presenter subunit. This guy is a catalytic because the actual catalytic site is inside the bigger part of the 50S subunit of the ribosome. So the small subunit only has really narrow channel for the threading of the mRNA molecule through the ribosome. And again, that's held together by the rRNA, the 16S RNA that's inside the 30S ribosome. So this narrow channel prevents any sort of secondary structure in the mRNA and allows for very tight control of the mRNA so that kinks can be introduced so that the codons can be partitioned into the triplicates, triple nucleotide codons that are necessary for the proper reading of the genetic code.